Hello students, and welcome to your lecture for Chapter 5 on the fundamentals of human energy transfer. I do want to point out the chapter objectives. They're really important to understand and be aware of both before and after the chapter, mostly before so that you can have an idea of the information that you should be keying up on, as well as after the chapter. It is a good checklist to go over to make sure that you've understood and picked up the most important information. So at this time, before we get started on the lecture, I do want to point out your objectives. You have three major portions of Chapter 5, and the objectives are listed in no particular order. But I just want you to be aware for your chapter outlines that you will be doing that you're going to split up your outline according to the three different parts. So let's go over the objectives. We're going to talk about the law of thermodynamics and how it's related to energy balance and biologic work. We're going to talk about potential versus kinetic energy, what it is. We'll give some examples. We'll talk about exergonic and endergonic chemical processes within the body. We'll talk again about second law of thermodynamics and talk about practical applications in regards to exercise science. We'll talk about forms of biologic work. You will need to know those three particular forms. We're going to jump hardcore into talking about role of enzymes and coenzymes in bioenergetics. We'll look at high energy phosphate bonds and discuss their contributions into powering biologic work, as well as outline the process of the electron transport oxidative phosphorylation through the electron transport chain. Sorry, this is some wordfuls for me this evening. Final chapter objectives talk about explaining oxygen's role in energy metabolism. We'll look at anaerobic energy release and how oxygen is not involved in that particular process. We'll discuss lactate formation. If any of you have heard of the term lactic acid, this is commonly mistaken for lactate, or maybe lactate is actually commonly misinterpreted or confused as lactic acid. There's a huge difference. We'll talk about that formation during increasing intensities of physical effort. We'll talk about the Krebs cycle, otherwise known as the citric acid cycle during macronutrient catabolism. We'll talk about adenosine triphosphate yield from the catabolism of our macronutrients, and then we'll kind of describe a little bit more in depth about how fats burn in a carbohydrate flame. So if none of these really make sense to you right now, that's because you probably haven't read the chapter yet. If you have read the chapter and now you're listening to this lecture, some of these things should make more sense to you. But let's go ahead and get started with your lecture. So Chapter 5 really looks at the body's capacity to extract energy from those food nutrients and transfer it to the skeletal muscles' contractile elements, which really determines the capacity for physical movement. So that's a big reason why we started off with a lecture in Chapter 3 regarding carbohydrates, proteins, and lipids, is they are kind of the starting foundation for our discussion into getting our muscles to move and then developing into some type of movement like athletic performance. So this energy transfer occurs through thousands of complex chemical reactions that require a proper mixture of macronutrients and micronutrients continuously fueled by oxygen. So I want to take an opportunity to kind of differentiate the terms aerobic and anaerobic in nature, because aerobic describes oxygen requiring energy reactions, where in contrast anaerobic energy reactions generates energy rapidly from the chemical reactions without oxygen present. You'll hear more of that in just a little bit. But please take note, the anaerobic and the aerobic breakdown of ingested food nutrients provides the energy source for our bodies to synthesize the chemical fuel that powers all forms of biologic work possible. So this chapter really presents an overview of the different energy forms and also factors that impact energy generation. We also discuss how the body obtains energy to power its diverse functions, and a basic understanding of carbohydrate, fat, and protein catabolism provides the basis for much of the subject matter content of this particular course in exercise physiology. So having a knowledge about human bioenergetics guides a practical basis to forming sports-specific training regimens, recommending activities for physical fitness, for weight control, and also for advocating prudent dietary modifications for specific sport requirements. So you're slowly beginning to understand how truly foundational the information within a course such as exercise physiology is in terms of your professional careers. Let's jump right into part one that talks about energy, the capacity for work. So I really want to talk about how the fact that energy suggests a very dynamic state related to change. So the presence of energy truly emerges only when a particular change occurs. So this becomes important for us to talk about the first law of thermodynamics. And that states that energy cannot be created or destroyed, but it's only transformed from one state to the other without actually being depleted. In the body, chemical energy stored within the macronutrients bonds doesn't immediately dissipate as heat during energy metabolism, and instead a large portion remains as chemical energy, which the musculoskeletal system then changes into mechanical energy and then ultimately into heat and energy. So when you guys think about it, when we ingest or when we take in our macronutrients, our carbohydrates, our proteins, our lipids, our body doesn't just automatically process that immediately for use. It basically assesses what is present within the body, what storages are depleted or near being depleted, and it takes the right nutrients and stores them for the appropriate moment when it is time to perform some sort of physical activity. So this kind of goes into the next idea of the conservation of energy. So we've got the mechanical energy, it's stored within the bonds, doesn't immediately dissipate. During energy metabolism, again, the large portion of what we ingest is stored. It's saved as chemical energy, which then our musculoskeletal system changes into mechanical energy when it's time for us to, let's say, get up and go. So this leads us to our next kind of definition and topics relating to potential and kinetic energy. This is basic science that's learned initially, I believe, in the elementary school levels, and then it's revisited in middle and again in high school. But the potential and kinetic energy together combined constitutes a system's total energy present. So when we release potential energy, this transforms basic ingredients into the kinetic energy of motion itself. So if you look at the picture here of the waterfall going over the dam, you can see that the water at the top of the dam represents that trapped potential energy, and when the water actually begins to fall over the dam, it actually dissipates and turns into kinetic energy as it really kind of does that cascading effect. And then the water at the bottom of the dam represents that lower potential energy. This also shows a scientific point that we have things of a higher gradient tend to flow to where there's a tendency to have a lower potential gradient. So we've got, again, water at the top of the dam is the potential energy. It's not in motion yet, and once it begins to cascade over the side of the dam, then it becomes kinetic energy in process. So we've got this bound energy in one substance that directly transfers another substance to increase its actual potential energy. And so this is an example of providing the required energy for biosynthesis. And when we talk about biosynthesis, when we talk about specific building block atoms of carbons, hydrogens, oxygen, nitrogen, they become activated and join other atoms and molecules to synthesize important biological compounds and tissues. Some newly created compounds provide structure, as in bones, or cells in the outer lipid-containing plasma membrane, things like that. The synthesized compound, such as adenosine triphosphate, or ATP, that you're going to hear a lot of starting this chapter, as well as phosphocreatine, provide the main source of a cell's energy requirements. Remember that, because that's going to be very important here in just a little bit. So let's talk about the exergonic and endergonic reactions that take place within a biological system. Definitions for you here: exergonic reactions are any physical or chemical processes that releases energy into its surroundings. This kind of represents the downhill process, or the process of decline in free energy. The way that I like to remember the difference between exergonic and endergonic is exergonic sounds a lot like exit. Or to go out of. So that might be a prefix that you could help you remember the definition. Endergonic is the opposite. This is the chemical process that actually stores or takes in or absorbs energy. And this is obviously the opposite of exergonic in that it represents the uphill process. It proceeds with increase in free energy available for biologic work at a later point in time. This leads us to our next concept in regards to the second law of thermodynamics, in that the transfer of potential energy in any spontaneous process always proceeds in a direction that decreases the capacity to perform work. It also applies to the tendency of potential energy to convert to kinetic energy with a lower capacity for work. So that's what's referred to as the second law of thermodynamics. Because ultimately, all potential energy in a system degrades or degradates to an unusable form of kinetic or heat energy. So an example of this would be the potential energy that's stored within a battery, and over time, slowly that energy downgrades or degrades to unusable form. Let's discuss some examples of energy conversions within living cells. For example, photosynthesis and respiration represent the most fundamental examples of energy conversion. Photosynthesis is an endergonic process which is powered by the sun's energy. The pigment of chlorophyll is actually located within a leaf's large organelles, and the chloroplasts absorb radiant or solar energy to synthesize glucose from carbon dioxide and water, with oxygen flowing freely into the environment. The plant also converts carbohydrates to lipids and proteins for storage as future reserve for energy and growth. Animals then ingest plant nutrients to serve their own energy needs. So truly, in essence, solar energy coupled with photosynthesis powers the animal world with food and oxygen. This picture that you see here is the opposite of an endergonic reaction. It is referred to as exergonic chemical or energy conversions. And this actually illustrates the exergonic reactions of respiration, which is the reverse of photosynthesis, as a plant stored energy is recovered for biological work, shown in the bottom three panels that you see here. So these three exergonic reactions happen within the presence of oxygen, and our cells extract the chemical energy stored in the carbohydrates, the lipids, and the protein molecules, and then actually utilize and oxidize some of those reactions and end up giving off heat. So this kind of leads us to our next concept then in regards to biologic work within humans. So you can see that the mechanical work requires the use of ATP, which is the energy currency of our muscles. So anytime we pick up a pencil, pick up a book, we are performing an exergonic process called mechanical biologic work. There's also chemical work, which synthesizes cellular molecules. For example, when we are converting glucose into glycogen. So when we have free-floating glucose within the blood and it's being stored within the muscle or the liver, and ATP is present there, our body is performing chemical work. Another example would be tissue repair. After a really hard resistance training bout, when we need to repair the small micro tears that have occurred within the muscles, that is a form of chemical work to synthesize or resynthesize tissue that was minorly damaged. And then the third example of biologic work in human is transport work. And this concentrates various substances in the intracellular and extracellular fluids within the body. So the example that you see here is the ATP being converted to ADP with the help of an extra phosphate, and the sodium phospholipid bilayer within the cell. So if you missed all that, I just kind of pulled all that information out and put it in this particular slide for you. It is listed for you in your textbook as well. So what affects bioenergetics? There's a bunch of them. Let's talk about the enzymes first. Enzymes are highly specific, large protein catalysts which can either speed up or reverse the rate of the chemical reaction. Enzymes are never consumed or changed. They are constantly preserved within the body, and they go on being used and used and used over and over again. Another factor that affects bioenergetics are reaction rates, so specifically the operation rate of the enzyme. Two things that majorly affect the reaction rates are the pH of the blood and the temperature of the body. These can dramatically impact enzymatic activity and either impede or provide the best environment possible for enzymatic activity to take place. The mode of action also affects bioenergetics, so this is how an enzyme interacts with its specific substrate, and we'll talk more about kind of that lock and key mechanism that really allows the enzyme to perform the action. And then we also talk about coenzymes as another factor. Coenzymes sounds kind of funny enough because enzymes are supposed to be catalysts themselves, but coenzymes assist the enzymes in their functioning. So these are complex non-protein substances. Remember, enzymes are protein catalysts. The coenzymes are non-protein, and these facilitate enzyme action by helping bind a substrate with its specific enzyme. So not only are enzymes important in affecting bioenergetics, but their friends, the coenzymes, are also really important in assisting in that binding of certain substrates. So you can see here an example if you forgot work from your previous biology or chemistry class, or maybe even an A&P. This is kind of a good graphic or a good image to help you picture the reactions that are present within biological work. So you can see we've got a substrate that matches with an active site of an enzyme. Remember, this is that lock and key mechanism where specific enzymes can only match their perfect active site. If they don't match, they literally just bounce off the surface and they continue floating on until they can perform kind of that lock and key mechanism. And so when they do join together, you see the enzyme substrate complex, and you've got the lock and key that is fit perfectly, and then it's hydrolyzed with water, and the enzyme substrate complex splits, and then it yields the new product, with the enzyme never being consumed, never being reproduced. It is simply something that is used over and over again. It becomes available for interaction with another substrate. And again, enzymes can also function with the assistance of coenzymes. And again, pH and temperature are very, very important in chemical reactions within the body. Let's take an opportunity before we move on to part 2 of chapter 5 to go over the most important concepts from part 1. And we're going to go straight down the bulleted list. Be aware for your first quiz, the body transforms energy from one form to another as physiological systems undergoes change. Remember, potential plus kinetic energy together combined make the total energy present within a system. Remember that exergonic reactions release or allow heat to exit, while an endergonic reaction brings in energy or traps heat. The total energy in a system remains constant. If there's a decrease in one form of energy, it means that there's an increase in another form present. Know the 3 forms of biological work. So we talked about chemical, mechanical, and transport work. Be aware that enzymes accelerate our reactions rate and are greatly affected by pH and temperature. And our friends coenzymes help the enzymes through facilitation of enzymatic actions. So at this point, if you are still confused with some of the major concepts or you missed something, feel free to pause the video at this point and go back and either look at your notes or look at your outlines for Chapter 5 for Part 1, or reread the text for more clarification. Or you can also send me emails with questions. At this time, we're going to go ahead and jump in with part 2, looking at phosphate bond energy. When we talk about phosphate bond energy, we understand that the human body receives a constant supply of chemical energy in order to survive. Yet the energy from the food that we consume doesn't release suddenly because our body can't directly harness that heat energy. So when we talk about energy being released, that's considered food oxidation. So when we talk about the changing of the macronutrients, or when our body is utilizing those macronutrients, that's referred to as food oxidation. So rather, we've got enzymatically controlled reactions that occur within the cell in order to extract the chemical energy that's trapped within the macronutrients molecular bonds. And there's 2 really good reasons that benefits the human body. First and foremost, it preserves energy. It saves it for when we have a true need for that. Let's say that we're in a fight or flight situation and the building is on fire and we have to get up and go. Our bodies can extract that energy from our cells through enzymatic reactions to aid us in being able to run screaming with our hands waving in the air out of the building. Another benefit to these extraction of this chemical energy really enhances the inner transformation efficiency, again, to aid us when our performance is required to be at its best. So fortunately, the body can directly use chemical energy for many forms of biologic work, ranging from the ciliary movement within the lungs, nerve impulse transmission to signal the heart to actually pump and beat, finger movements for something as delicate as playing the piano, and the movement of food through the small intestine and the colon. When you hear the words adenosine triphosphate, this is our ATP, this is the actual energy currency that provides us this free energy for all cellular functions that take place. You will hear ATP frequently within your degree, bachelor's degree program in exercise science. And so there's 2 major energy transforming activities that take place within the body. First, we've got forming and conserving ATP from food's potential energy, but also using that extracted energy from ATP to power all forms of biologic work. So let's take a look at this video known as the energy transfer chain. Let's see if it's going to come up. Okay everyone, so I tried to pull the video up and it's doing something really funky where you're basically hearing the voice double over itself, and it's really frustrating. So instead of me pulling it up and showing it to you within this particular lecture, I'm just going to point out to you that you do have the capability through that link provided to you on Blackboard that can get you into, I believe it's under program resources, it's called The Point. And if you log in with the code at the beginning of your textbook, you will have access to specifically all of the chapters, but specifically for this lecture, Chapter 5, and all the videos that go along with Chapter 5. So if you want to pause the video right now or the lecture and go over to the link on Blackboard so you can view the video on the energy transport chain, it's basically summarizing and showing a very basic graph in regards to the energy transfer chain that you're going to hear within the rest of the PowerPoint anyway. So at this time we're going to go ahead and go on to the next slide. So what is ATP? If this is the actual cell's energy currency, what is it? What's the chemical structure? So it's actually made up of adenosine, and then the second part of it is triphosphate. So what you can see within this are the 3 high energy phosphates that are linked together with these high energy phosphate bonds. So there's potential energy here, and there's a potential energy here as well. If you watch the video on the energy transfer chain, it talked about the cleaving of the final phosphate bond, which would then remove one of the phosphate and turning adenosine triphosphate into adenosine diphosphate. So if you're remembering your prefixes here, Di represents 2 and Tri represents 3. So at this point, we've got our high energy bonds that are present with an adenosine triphosphate, and when that energy is released is when this is cleaved and the phosphate bond is released, and we're then left with adenosine diphosphate. Well, why is this important? Why are we discussing ATP and ADP? When we have that release of that final phosphate, or that final phosphate bond is cleaved or broken off, what happens is that is a release of free energy to the system. So when each mole of ATP is degraded to ADP, this liberates approximately 7.3 kcals of free energy for biological work. So don't get scared by the formula that you see below. It's basically a picture form or a math formula representing exactly what we've said. We have ATP. Water comes in and acts as an enzyme or a catalyst to cleave that final phosphate bond, and so that's why you see the ATP ACE, and that leaves us then with adenosine diphosphate plus an extra phosphate floating around in the system. So this is kind of like an energy receiver, energy donor cycle that represents a cell's two major energy transforming activities. The first is to form and conserve ATP from food's potential energy. And the second is to use extracted energy from ATP to power biologic work. So the splitting of ATP takes place without oxygen. Usually that means it's anaerobic in nature. And this generates energy for rapid use for all biological activities. For example, for us specifically, we study muscle action. So this is sort of a backup power source in excess of aerobic energy production, therefore with oxygen. So I want you guys to think about everything that you do in your daily life that doesn't really require a whole lot of thought, a whole lot of effort involved. Lifting a pencil, turning a book page, walking from your car, from the parking lot into class. It could even be spiking a volleyball or jumping up and down. This is all examples of anaerobic energy production because your body does not have to wait for oxygen to be present within the cells to power these type of activities. When we're talking about biologic work, this is a great picture that kind of gives you examples of all the different type of work that our body does on a daily basis. So in the middle of the graph, you can see the adenosine and the triphosphate and the bonds present there. It transfers and powers nerve transmission, our muscle action, circulation, tissue synthesis, where we have our amino acids that combine with ATP to formulate proteins. It helps us with our digestion, and it also helps us with hypothalamus and glandular secretion right here in the upper right hand corner. So truly, this is another picture of why we call ATP the energy currency of the cell. It literally powers all biological work that takes place. Without ATP, we'd be in some serious trouble. So why do we have to wait for oxygen to be present to switch from anaerobic to aerobic energy production? And that's because of a couple different reasons. ATP is something that is extremely heavy. The molecular structure of ATP, if we were to store enough ATP for everything that we would need to do during our day, we would not be able to stand up or get out of our chair. It would be that heavy. So our body can only store a certain amount of ATP at a time, to the tune of only about 80 to 100 grams of ATP, or about 3.5 ounces. This is an advantage because of the molecule's heaviness. And so because it is so heavy in nature, because we have a limited supply of it, our bodies have to resynthesize ATP continually to match its rate of use. And when we talk about the resynthesis or the regeneration of ATP, it's produced in different ways. Some of it is produced in the cell's cytosol or the cytoplasm, and this is where the anaerobic reproduction takes place. It's also referred to as anaerobic glycolysis. So you can see that in our picture here on the right hand side. It utilizes phosphocreatine It bumps up our glucose and glycogen storages. It utilizes glycerol, some deanimated amino acids, and boom, we've got some ATP created to power our biological work. So this is for activities, and I'm getting a little bit ahead of myself, but this is for any activities that we do on a normal basis. We're not, needing a whole lot of energy to pick up our pencils, to type on the computer, to talk on the phone, things of that nature. This is kind of our backup system. The system that really takes over after activities last longer than about 90 seconds is aerobic glycolysis. This is also referred to as the Krebs cycle, the citric acid cycle. It's all this, excuse me. Sorry guys, I got a little frog in my throat there. The Krebs cycle, the citric acid cycle, is all different names for the same process. And this is called aerobic glycolysis. So that means it happens with the presence of oxygen. So when we talk about the CAC, or the Krebs cycle, this takes place within the mitochondria of the cell. If you guys remember, the mitochondria is the powerhouse of the cell. This is where oxygen needs to be present for ATP to be reproduced and give us biologic work. So you have probably heard me talk about the fact that when we're walking, talking, typing, doing schoolwork, doing classwork, things of that nature, we don't really need oxygen to be present. This is called the first energy system that's present, called the ATP-PCR cycle, and this is our immediate energy system that's utilized to get us powered up and ready to go. So we utilize phosphocreatine. It's also called as creatine or creatine phosphate, but it's called the ATP-PCR cycle. PCR is an additional stored high-energy phosphate that helps in the reproduction of ATP. So it provides some energy for the resynthesis to take place. It releases a considerable amount of energy when the bond splits between the creatine and the phosphate bonds. So that's what you can see happening here in your picture. Cells are able to store considerably more PCR than ATP, and that's helpful when it comes to the time when we need to replenish the ATP anaerobically. And the mobilization for energy takes place almost instantaneously and therefore requires no oxygen. So you might also have your textbook refer to the ATP-PCR system as the immediate energy system. So this is for short duration work. This could also be for very powerful, very explosive work to the tune of up to maybe 15 seconds. So this is our anaerobic source of phosphate bond energy, where the energy is liberated due to the hydrolysis, which is a splitting of the PCR and rebonds with our ADP and phosphate to form ATP. You have a really good picture that kind of exemplifies this for you. So if we have ATP that's already stored within our system, we have a quick reaction that requires to jump out of our chair for something. That ATP is split with an enzyme to release 7.3 kcals of energy for whatever biologic work that our body needs to perform. And then we are left with ADP, two phosphates attached to adenosine, with an extra phosphate floating around. And so we're left with this product. And to resynthesize ATP, we have phosphocreatine that combines with adenosine diphosphate to reformulate, take that phosphate off of the creatine and put it right here at the end of the adenosine diphosphate to recreate adenosine triphosphate. So again, the first energy system, the immediate energy system that our body taps into when it's time for us to walk, talk, breathe, push a pencil, is the ATP-PCR immediate anaerobic energy system. So this slide goes a little bit more into the ATP-PCR breakdown. That breakdown, or that energy requirement of the body, sustains all-out efforts lasting about 5 to 8 seconds. It's a very, very short window. So when you see an Olympic sprinter, when you see an Olympic swimmer, they begin to actually slow down at the end of a race instead of maintain or even increase that speed because of the fact that their high energy ATP PCR system is basically tapping out all of the ATP that's present. The winner is the one that most effectively supplies and uses a limited quantity of phosphate bond energy within their system. So again, the first energy used in the human body is the ATP PCR system, and that's why it's called the immediate energy system. If you are in any type of physical activity that requires movement or power or performance beyond 8 seconds, the body then requires an additional energy source to sustain the activity, and we tap into the next energy system. We're going to skip this because this is something that we might do tomorrow together in class. Let's talk about identifying predominant energy systems. This is the basis for developing effective training programs. So first we've talked about the ATP PCR immediate anaerobic energy system. High energy output sports that require short, powerful bursts of energy for only a limited short amount of time, like football, track, maybe baseball, all rely exclusively on energy transfer from the intermuscular high energy phosphates. So it makes sense then, as someone that would train or program exercise for these type of athletes, that we develop this type of energy system to create effectiveness for that particular sport, even for that particular position for that athlete within that sport. So we as professionals need to understand how we can optimize different energy systems' power output capacity. I know sometimes we sit in class and go, why do I need to know this? And this is a perfect slide to kind of explain to you why we study the macronutrients, what those macronutrients do, and how they affect the energy that's present within our system. So when we talk about the ATP PCR immediate energy system, understand that not everyone needs to have these short, quick, explosive bursts of power. Can you guys think of any sports that are enduring in nature, that are very long, that are very time-consuming, that require intermittent bouts of moderate and vigorous intensity activities? Think about it for a second. Has anyone thought of maybe marathon running or cycling or participating in the Tour de France, even sports such as basketball and soccer, where you're continuously running, but you have some short bursts of power when you have to sprint up the field, and then you stop and recover and you gain your position again, and then you sprint again. So those particular types of activities that last longer than 8 seconds or even 30 seconds, ladies and gentlemen, utilize a different energy system. Let's discuss phosphorylation, because this is where we talk about chemical bonds actually transferring the energy. So phosphorylation is when work occurs when the compounds relatively low in potential energy juice up from an energy transfer through these high energy phosphate bonds. So if you remember that picture of the adenosine and the 3 phosphates that are present, ATP serves as the actual energy transfer agent. The phosphate bonds actually trap a large portion of the original food molecules as potential energy. And then ATP transfers this energy to other compounds to raise them to a higher activation level. So this energy transfer through the ATP phosphate bonds to other compounds and produces higher activation level. This is called phosphorylation. And it does this through the assistance from the oxidation or the biological burning of the molecules. So an oxidation is actually called the transfer of electrons, and a molecule becomes reduced if it accepts electrons, and a molecule becomes oxidized if it gives up an electron. This is something that you do need to remember the process of oxidation, and you'll see a slide here come up in a little bit that reminds you of the acronym oil rig. So when we're talking about the transfer of electrons, this actually helps refuel and cause the phosphorylation process to occur, and it's through the transfer of electrons. So again, if a molecule is reduced, it accepts or gains electrons. And again, if it loses or gives up an electron, it becomes oxidized. So this is called oil rig. Oxidation, loses, reduced, gains. And this constitutes the mechanism for energy metabolism. So again, oxidation is this biologic burning of macronutrients. It provides the energy required for the phosphorylation to take place. It happens in the inner lining of the mitochondrial membranes. Specifically, we're involving transfer electrons from NADH and FADH to a molecular oxygen, which then releases and transfers chemical energy to combine ATP from ADP plus a phosphate ion. 90% of ATP synthesis takes place in the respiratory chain by oxidative reactions coupled with phosphorylation. So that's kind of the bottom line of what we're talking about. Remember, oxidative reactions is the gaining or losing of electrons, and phosphorylation, again, let me back up for you, is when we have chemical bonds being transferred through energy. Okay? So I don't want you to get too scared by the next couple of graphics that come up, because what they do is describe to you the electron transport chain, and they show the oxidation, the gaining and losing of electrons as they are passed down this electron transport chain to aid in phosphorylation. So this is a picture of oxidizing hydrogen and accompanying electron transport that reduces, remember, gains oxygen and forms water. So the oxidation of hydrogen and the subsequent phosphorylation that occurs looks like this big scary formula. Don't get intimidated by this formula. What I want you to do now is pause this particular video and go to your Program Resources tab on Blackboard. Access the point, which is your online component of your textbook, and watch the video on electron transport. Now that you've had an opportunity to view the video, this is just something I put in the PowerPoint to assist you in your studies, to remember the difference of oxidation, what it means to gain and lose electrons. So again, this is my acronym that I explained earlier. Oil is oxidation, involves losing or losing electrons. Rig, R-I-G, means if there's a reduction, it involves gaining an electron. Sometimes people get that backwards because reduction and gain doesn't quite go together. But you will need to know that. So these are good pictures, again, good graphics of harnessing potential energy. These are pictures of kind of the electron transport chain. So this particular picture that you see on the left-hand side shows us our higher potential energy, the kinetic engine energy happening as the waterfall goes down and through the turbines. And then it results in lower potential energy. So that is comparative to the picture that you see here on the right when we talk about in the body. So again, the left-hand picture is like an industry picture of what happens with mechanical work, maybe with machines, and now we're comparing it to the electron transport chains of the oxidation process of gaining or losing electrons in the human body. And the main point that I want you to get from looking at the oxidation process, ladies and gentlemen, is a couple different things. First, please don't get scared off by the technicality of your textbook and of these graphics. That's one of the reasons that I create these lectures for you. This particular picture is showing you something very good that I hope that you get out of this. Through the oxidation process of gaining or reducing electrons, so gaining or losing electrons, ATP is released out through the body. And that's a good thing because that gives us the power to do biologic work. So we have higher potential energy here. We have the NAD and the FAD that's very unnecessary in order to aid in this gaining of electrons or losing electrons down through the electron transport chain. That process, again, produces ATP at certain intervals, giving us the ability to perform biologic work. So that's the first thing I want you to understand about the electron transport chain. The other thing that I want you to get out of this graphic is that oxygen is the final receptor at the end of the electron transport chain. And that's stated for you in a couple slides here. So oxygen's role in energy metabolism. This is kind of picking up along with the graphics that we just talked about. Continual resynthesis of adenosine triphosphate happens during coupled oxidative phosphorylation of macronutrients. And it has three prerequisites. There has to be the availability of the reducing agents NADH or FADH. And again, that's why you see these here on the left-hand side of your screen. There also has to be a presence of terminal oxidizing agent as oxygen. So that's why, again, we said that oxygen is the final receptor in the electron transport chain. The third prerequisite is that there has to be a sufficient quantity of enzymes and metabolic machinery in tissues to actually make the energy transfer reactions go at the right sequence and at the right rate. So I know that's a lot of information, but that actually completes part 2. So take an opportunity to read through. These are excellent bullet points that you should maybe study and be aware that will help you be successful for your chapter outline, for completing your chapter homework, and ultimately studying for your quizzes. I'm not going to take the time to read all of these to you because it's simply a review, but these are some of the most important parts from part 2. The final component of Chapter 5 is Part 3, and this goes into detail in regards to the energy released from food. Our body depends on 6 macronutrient fuel sources to supply substrates for oxidation and ATP regeneration. So before we go on, let's review. This whole chapter is about bioenergetics, or the energy systems present that allow biologic work to take place. We have discussed the first immediate energy system and its processes. So we've talked about anaerobic the ATP PCR immediate energy system that operates without the presence of oxygen. This information that we're introducing now is leading us up to the next 2 energy systems that our body utilizes whenever our physical activity lasts longer than 10 to 12 seconds. So if your brain is a little fried at this point, stick with it. Stick with me in your lecture. I understand that this can be some overwhelming information, but I wanted to give you an idea of where this chapter is actually going. So again, talking about energy released from food, we depend on triglycerols, specifically glycogen molecules that are stored within our muscle cells. We also depend on our blood glucose levels to supply kind of more immediately what our cells need. We depend on free fatty acids. We depend on the amino acids present in the intermuscular and liver-derived carbon skeletons. Anaerobic reactions in the cytosol in the initial phase of glucose or glycogen breakdown. So that's referring to the ATP PCR immediate energy system. And then also it depends on something we just talked about in regards to the phosphorylation of ADP by the phosphocreatine, as well as creatine kinase and the adenylate kinase enzymatic control. So again, I like pictures, and I thought this was an excellent graphic to share with you that looks at the fuel sources that supply substrates to regenerate ATP. So you will hear us frequently talk about the glucose within the blood and the stored liver glycogen, deanimated amino acids. A lot of times when people take branched chain amino acids, that's kind of what they're referring to or thinking that they're doing to help with the replenishment of ATP. We've got intermuscular energy storage within the muscle, and also we've got triglycerols and fatty acids within the adipose tissue. All of those enter into the bloodstream and affect the citric acid cycle that you can see here. Again, another name for that is the Krebs cycle. That allows the electron transport chain to happen, which oxidizes or reduces electrons in order to formulate, here it is, kind of the whole point of our lecture, the ATP, in order to allow biologic work to continue. So we're going to focus on carbohydrate energy release first. And the discussion of this particular nutrient begins with carbs because of a couple different reasons. It's the only nutrient whose potential energy generates ATP both with and without oxygen. Carbohydrates supply about half of the body's energy requirements during light to moderate aerobic exercise. This is why when athletes say they're going on a carbohydrate-free diet, we need to kind of have our red flags go up and really begin to listen to the athlete and try and understand, where did they get this information? Why is this athlete communicating that they feel there's a need for them to be on a carbohydrate-free diet? It can be very detrimental to their health in terms of energy intake and energy output. And it also can be extremely detrimental to their athletic performance as well. We also look at carbohydrates first in energy release because of the processing fat for energy requires some carbohydrate catabolism. So a breakdown of carbohydrates can affect the processing of the body's fat. Aerobic breakdown of carbohydrates for energy occurs at about twice the rate as energy is generated from the lipid breakdown. So it's a very dependable, it's a very important energy source for our body. And to function optimally, our central nervous system requires an uninterrupted supply of carbohydrates because of the fact that glucose is so important as a fuel to our brain. So what is the primary function of carbohydrate? It is to supply energy for cellular work. It represents the only macronutrient whose potential energy generates ATP aerobically. Complete breakdown of one molecule of glucose yields a maximum of 686 kcals of chemical-free energy available for work. And the oxidation of one glucose molecule in skeletal muscle yields about 32 ATP molecules. Don't panic about the formula that you see at the bottom. It just kind of reiterates what you've seen on this page. So when we talk about the next energy system that our body taps into after the ATP PCR cycle has been completely maxed, then we start talking about anaerobic versus aerobic reformation of ATP. So glycolysis is referred to as carbohydrate breakdown. Aerobically, this is a slower process resulting in substantial ATP formulation. So the second stage of carbohydrate breakdown converts something called pyruvate to acetyl-CoA, and then we drop down and start beginning our process through the citric acid cycle, which again requires the presence of oxygen. If we have anaerobic glycolysis, this is relatively rapid in nature, but it supplies us only with limited ATP production and produces the end product of lactate, which is not aerobic in nature. So this is setting us up for the next energy system that's present. Let's go back. The first energy system that we tap into is the ATP PCR cycle. It's our immediate energy source and it occurs without oxygen. It also only lasts for a certain amount of time, no more than about 8 to 10 seconds, and then our body has to tap into a different process to supply the regeneration of ATP. So let's talk about anaerobic glycolysis, which is referred to as the moderate to moderate intensity work. This is the actual first stage of glucose degradation. It only generates about 5% of ATP. It is a crucial energy system during maximal all-out physical activity for up to about 90 seconds. So once we've tapped out our ATP PCR energy system, then we have a dimmer switch that kind of turns that particular energy system down, but turns up the anaerobic glycolysis process for replenishing ATP. It occurs in the cytoplasm of a cell still, which means that it's occurring anaerobically without oxygen. So we have carbohydrate energy release that aids in this next particular energy system. So that's why we call it the rapid anaerobic glycolysis. Again, don't get confused or scared by the graphic that you see included here on this particular side. What it does is show you the series of 10 enzymatically controlled reactions that creates 2 pyruvate molecules from the anaerobic breakdown of glucose. So you can see pyruvate is the end product of this whole process of rapid anaerobic glycolysis. So that's what I want you to remember. The name of this picture and the end products with the possible presence of lactate forming when the NADH oxidation does not keep pace with its formation of glycolysis. So let's highlight and let's talk a little bit more about what this lactate formation is when pyruvate is present. Let's talk about lactate formation for a second, and I don't want you to confuse lactate with something called lactic acid. Lactic acid is also referred to as milk acid. It is not something that is used interchangeably with lactate. Lactic acid, an acid formed during anaerobic glycolysis, quickly dissolves to release a hydrogen ion. So it's very, very different in nature. What I do want to do is kind of clarify how lactate forms and also why it comes from pyruvate. So we have sufficient oxygen that bathes cells during a light to moderate levels of energy metabolism. So and relatively light to moderate physical activity. Electrons are stripped from a substrate and carried by NADH to oxidize with mitochondria to form water when oxygen joins with them. And in a biochemical sense, a steady rate exists because hydrogen oxidizes at about the same rate that it becomes available. So this condition of aerobic glycolysis forms pyruvate right here as our end product. But in strenuous physical activity, when energy demands exceed the oxygen supply or the utilization rate, the respiratory chain cannot process all of the hydrogen joined to NADH. A continued release of anaerobic energy in glycolysis depends on the NAD availability for oxidizing this very large reaction. Otherwise, the rapid rate of glycolysis actually comes to a halt or a complete stop. So during rapid or anaerobic glycolysis, NAD frees up as pairs of excess non-oxidized hydrogens combine temporarily with pyruvate to form lactate and then catalyzed by the enzyme lactate dehydrogenase in the reversible reaction that you can see present here in figure 515. Also, I want to state that lactate should not be viewed as a metabolic waste product. It's actually very useful, and it accumulates with intense activity and is a valuable chemical energy source. And so I want to talk to you a little bit about the Cori cycle as well. So this lactate that's produced in the fast-twitch muscle fibers and other tissues actually circulates to aid in the conversion of pyruvate. And when this occurs, pyruvate then converts to acetyl-CoA for entry into the citric acid cycle for aerobic energy metabolism. Again, do not make the mistake of considering lactate as lactic acid. It's actually something that's very useful. The Cori cycle, or the lactate shuttle system that we have present within our body, takes what's present within the muscle, transports it through the bloodstream back to the liver to reassist and use that byproduct in the form of reforming ATP. Oops, let me back up that one. So that's what this picture then shows, is the slow aerobic glycolysis through the reactions of the Cori cycle that happens in the liver to synthesize glucose from lactate. You have several videos, again, on the point in your program resources tab on Blackboard. This would be an excellent time to pause the video and to go watch the videos on the Cori cycle and the alanine glucose cycle as well. This brings us then to the third energy system that's present within our body. So I want to stop again and go back. The first immediate energy system is the ATP-PCR anaerobic system that immediately replenishes ATP within the tune of about 8 to 10 seconds of activity. Once our activity continues beyond that point of time, then the next energy system that kicks into place is the anaerobic glycolysis that takes place again without oxygen. Its end product is either pyruvate or lactate, which then goes through the Cori cycle and it gets shuttled through the liver and aids in the reformation of glucose and ATP production. And that system typically lasts no longer than about 90 seconds of physical activity. So you're starting to hear and be able to comprehend different types of sports and what energy systems they predominantly rely on. So anything that's short explosive power up to the tune of about 8 to 10, maybe 12 seconds relies on the ATP PCR cycle. If the physical activity continues beyond 12 seconds, all the way up to about 30, 60, maybe even 90 seconds, our body is then ramping up the anaerobic glycolysis system, which typically ends in pyruvate or lactate. After any physical activity continues to occur beyond 90 seconds, then our bodies tap into and switch on the final aerobic metabolism system, which is considered the citric acid cycle or the Krebs cycle. There's kind of names used synonymously. And so that's the next portion of our lecture, is talking about pyruvate that actually enters into the citric acid cycle with the assistance of oxygen to replenish ATP. So phase 1 of the citric acid cycle occurs within the mitochondria. Remember, the previous two energy systems replenished ATP in the cytocell or in the cytoplasm of the cell. The citric acid cycle occurs within the mitochondria, the powerhouse of the cell, in order to generate hydrogen during the acetyl-CoA breakdown. So phase 1 looks like this. We have that whole process. Let me back up. We have this whole process that starts with carbohydrate breakdown. We have this whole different transfer of electrons, the electron transport chain that depend on NAD, NADH, and FAD. And all the way down here, we have all these processes that metabolically produce a little bit of ATP, but also require some ATP in order to be able to do this biologic work. And we end here with pyruvate or lactate. At this particular point, I'm going to fast forward because this is where we're picking up right here. If oxygen is present, then we drop into the formation of acetyl-CoA, which kicks off the reaction of the citric acid cycle. So this brings us to phase 2. There is significant ATP regeneration when the hydrogen oxidizes through this electron transport oxidative phosphorylation. Again, this is what's referred to as the electron transport chain. This is the energy system that produces the most amount of ATP for biologic work. So this is what one breakdown of one pyruvate molecule looks like. You will not have to know all these different small little steps in the introduction of the actoninase and the isocitrate, and I don't even know if I'm pronouncing a lot of these correctly. What I want you to understand, this breakdown of one pyruvate molecule produces 2 pyruvates, 2 acetyl-CoA molecules, and 2 ATP. The next couple slides talk to you about the production and the energy transfer associated with the macronutrients, so glucose, protein, and lipids. What I want you to do is really focus on these macronutrients and what produces the most amount of energy when they are catabolized. So I don't want you, again, to get too wrapped up in the graphics that you see here. The first macronutrient that our body depends on for energy regeneration is from our carbohydrates. Once that seems to be tapped out, once our glucose levels are low, then we start tapping into the energy release from lipids. And it's not always carbohydrates solely broken down first and then we tap into lipids. It's not in that manner, because stored fat is really the most plentiful source of potential energy. And you have a better understanding of that now after studying the Atwater effects, where we burn food in bomb calorimeters and understand the gross versus the net energy value present within foods. And we understand that lipids produce the most energy per gram of fat present, to the tune of 9 kilocalories. So there's 3 specific energy sources for fat catabolism. We look at the triglycerols that are stored directly within a muscle fiber. They're also close proximity to the mitochondria. We also look at circulating triglycerols, and we also then look at adipose tissue that provides circulating free fatty acids that are mobilized from the triglycerols in adipose tissue. Look at the amount of ATP molecules that are created during the breakdown of triglycerol molecules. We're talking 460 ATP molecules. Carbohydrates produce a net of 32 ATP molecules. And again, that makes sense according to the Atwater factors. So our body really uses the carbohydrates, the glucose, the glycogen that's stored first. And then when we have something like a marathon runner where their activity is lasting well beyond 60 minutes, our bodies heavily rely on these lipid sources as well. So this one might be a good time also for you guys to look at the video on the point called triglycerol breakdown, and it looks to explain the dynamics of fat mobilization and use. You can see the breakdown, and again at the bottom I just want you to focus on the fact that fatty acid breakdown can yield a total of 460 ATP. That's a lot, especially when you're considering that our body only stores 80 to 100 grams of ATP in one setting. So this is where we're talking about fats burn in a carbohydrate flame. Depleting carbohydrates decreases pyruvate production during glycolysis, and this reduces citric acid cycle intermediates, which slows the CAC activity. So this fatty acid degradation that occurs in the CAC depends on sufficient oxaloacetate availability to combine with acetyl-CoA formed during the B oxidation. And when carbohydrate levels decreases, an inadequate oxaloacetate level can also reduce that fat catabolism. This particular slide, ladies and gentlemen, read it, be familiar with it, but what I want you to understand is that our bodies depend on carbohydrates initially, and then lipids provide a lot of ATP production for us. I know that's zooming over this one. You can read it if you want. I'm not quite sure what kind of large questions will come of this. But you do see, again, the 460 ATP molecules that come from the fatty acid mobilization of lipids. So we talked about carbohydrates, we talked about lipids. Let's talk about the energy release from proteins. Protein is not a prime fuel source for athletes, and that's because of the way that it's digested and how slow it takes. It's also due to the fact that of the combination of the hydrogen ions that form the urea, which basically excretes protein out of our bodies. That's not to say, though, that protein isn't one of the macronutrients that our body depends on for ATP replenishment. It is, it's just not the first nor the second fuel source that your body depends on. It's actually one of the last. So this is an interesting picture. You can look over it. This is the picture that I really like that shows the dependency of the body on the different macronutrients that are present. And again, we've got our lipids, our carbohydrates, our proteins. Again, we've got our 9 kcals, 4 kcals, and 4 kcals per gram of the different macronutrients present. And you can see carbohydrates as a reason this is in the middle of the picture is because this is the primary fuel source that we depend upon. And so with the metabolism of these different macronutrients, you can see specifically with carbohydrates, it goes through an oxidation or a breakdown and can formulate pyruvate. And it also can end in lactate if certain molecules are not present. This can be shuttled back to the liver for replenishment of ATP in the Cori cycle. If we have pyruvate and oxygen is present, it is formed to combine acetyl-CoA, which then this is the gatekeeper, this is the key that can drop us into aerobic glycolysis, which is the final energy system that's utilized when activity persists longer than about 90 seconds. So if I have thoroughly confused you, what I would recommend doing is watching this video that I posted for you here. It is a great video. It's 4 minutes long. It's basically taken everything that I've said in regards to bioenergetics and simplified it. So if you can understand what's in this video, you have a good grasp of what bioenergetics means and how the human body powers physical activity when activity begins and lasts up to 10 seconds, persists to about 90 seconds, and then 90 seconds and beyond. So I want you to think of these energy systems as dimmer switches, because we never fully turn on or off one particular energy system. Our body simply transitions or shifts, or you've heard me use the example, uses a dimmer switch to turn one on and to dial one down depending on the length of physical activity. So I'm actually going to try and load up the video for you right now. Give me a second. Let me see if this is going to work. All right, ladies and gentlemen, my computer is acting a little funky, so I was not able to pull up the YouTube link for you. But what I would do is, if you can just type that into your web address, it is a great 4-minute video. It lays everything out that's much more basic for you in regards to the information from Chapter 5. So at this point, I would encourage you, the very end of your chapter has a great review of bullet points of the most important concepts from this chapter. I've also pulled those out for you at the end of this PowerPoint. Take some time, read it, make sure that if there are any particular bullets that you're still a little bit confused on, that's completely normal. I would expect that for your first go through for this information, but feel free to write some things down that if you have questions to bring to class, but use this as kind of a guide for studying for your quiz, for completing your chapter outline, and for doing your homework. And that is our lecture for Chapter 5.